David Baltimore, who passed away on September 6, 2025, was a scientific giant whose discoveries transformed biology. A Nobel Laureate, Baltimore's work in virology, immunology, and genetic engineering changed how we understand life. His most famous achievement, the co-discovery of reverse transcriptase, an enzyme that rewrote the rules of molecular biology. This breakthrough revealed that genetic information could flow from RNA back to DNA, overturning the central dogma and opening new frontiers in research. Baltimore's influence extended far beyond the lab. He led major institutions, most notably as president of Caltech, where he championed diversity and innovation. He was a public conscience for science, shaping ethical guidelines for new technologies and advocating for research during the AIDS crisis. Baltimore believed that great science required not just brilliant minds, but also strong infrastructure and a shared vision. He built environments where discovery could thrive, always mindful of science's responsibility to society. His legacy is one of relentless curiosity, leadership, and a commitment to using knowledge for the greater good. David Baltimore was, in every sense, a complete scientist, a discoverer, a leader, and a citizen of the world. For decades, biology taught that genetic information flowed one way: DNA to RNA to protein. In 1970, David Baltimore shattered this dogma by discovering reverse transcriptase, an enzyme that allows RNA to be written back into DNA. This finding revealed how retroviruses like HIV integrate their genetic code into host cells, changing our understanding of viruses and cancer. Baltimore's experiments were simple yet revolutionary. He showed that viral RNA could be converted into DNA, rewriting biology textbooks overnight. The discovery became the foundation for understanding and fighting HIV/AIDS, as reverse transcriptase became the target for life-saving antiretroviral drugs. Beyond medicine, reverse transcriptase became a crucial tool in biotechnology. Scientists harnessed it to create cDNA from RNA, enabling breakthroughs in gene expression studies and diagnostics. Techniques like RT-PCR, now essential for disease detection and research, stemmed directly from Baltimore's work. His insight didn't just solve a scientific puzzle, it empowered generations of researchers and clinicians. From the lab bench to the clinic, the impact of reverse transcriptase is felt every day. Baltimore's discovery is a testament to the power of questioning established truths. It changed the course of biology and medicine. One fundamental insight, and the world was never the same. As Caltech's president from 1997 to 2006, David Baltimore transformed the Institute's approach to biology. He elevated the life sciences, leading a major fundraising campaign that built the Broad Center for Biological Sciences, a hub for interdisciplinary research. Baltimore's vision extended to campus culture. He championed diversity, brought more women into leadership, and prioritized student life. He oversaw Caltech's role in NASA's Mars rover missions, expanding the Institute's reach beyond biology. Baltimore was both an administrator and an active scientist, running his own lab while leading the university. His leadership style combined intellectual rigor with accessibility, inspiring collaboration and innovation. Baltimore's presidency left Caltech stronger, more inclusive, and positioned for scientific leadership in the 21st century. His legacy at Caltech is one of bold vision and lasting impact. Baltimore knew science and society are inseparable. In 1975, he co-organized the Asilomar Conference, setting the first safety guidelines for recombinant DNA research, a model for scientific self-regulation. During the AIDS crisis, he was a leading advocate for research funding and national strategy, chairing influential committees that shaped policy. Baltimore's expertise reached beyond biology. He co-founded the Caltech-MIT Voting Technology Project, improving U.S. election systems. He was a voice of caution as gene editing tools like CRISPR emerged, calling for global dialogue and ethical restraint. Baltimore's public service showed that scientists must help guide society through new frontiers. He set the standard for responsible scientific leadership. David Baltimore's career was marked by a constellation of honors. He won the Nobel Prize at 37 for discovering reverse transcriptase, but his impact only grew from there. The National Medal of Science, the Lasker Tilde Koshland Award, and membership in the world's top scientific academies recognized his leadership in research and policy. Each award reflected a different facet: the revolutionary scientist, the institution builder, the public servant. Baltimore's achievements were not just personal milestones, they propelled science forward. His legacy is measured not by trophies, but by the progress he inspired. He remained a dynamic force in science until the very end. Beyond his scientific achievements, Baltimore was a legendary mentor. He nurtured generations of researchers, teaching them not just how to experiment, but how to think and question deeply. His lab produced leaders who shaped science worldwide. Baltimore's partnership with his wife, Dr. Alice Wang, a distinguished virologist, was a foundation of strength and intellectual companionship. Their home was a gathering place for lively discussion and community. Baltimore's curiosity extended far beyond biology. He was a lover of music, art, and culture, a true polymath. He moved seamlessly from scientific debate to conversations about Beethoven or fine wine. This breadth enriched his science and his relationships. He was demanding yet kind, approachable, and deeply invested in those around him. Baltimore's humanity was as profound as his intellect. David Baltimore's legacy is alive in every lab, clinic, and classroom touched by his discoveries. Reverse transcriptase remains a cornerstone of modern medicine and research, powering diagnostics and therapies worldwide. The institutions he shaped, Caltech, the Whitehead Institute, Rockefeller, continue to thrive on his vision of collaboration and innovation. His ethical leadership at Asilomar and in gene editing still guides science through new challenges. Baltimore showed that scientists can be both fearless discoverers and responsible citizens. He inspired a generation to pursue knowledge with curiosity and conscience. The world is better and science is stronger because of David Baltimore. His legacy continues to unfold, lighting the way for future discovery.